this topic, we're going to have a look at chromosomes and the cell cycle. So by the end of this topic, you should be able to answer the questions: what is a chromosome? What is the difference between heterochromatin and euchromatin? What is a gene and locus? What is a homologous pair of chromosomes? What is a karyotype? What is haploid and diploid? What is the cell cycle? And what is happening during each stage in the cell cycle? Okay, you all know that you've got a nucleus in a cell, and inside this nucleus are chromosomes. These chromosomes are made up of DNA, and the genes are the sections of DNA that are the units of inheritance, so they control a characteristic of the organism. So the gene for a particular characteristic is always found on the same position on the chromosome. So you get one gene coding for different features, and we call these alleles. For example, you have an allele for blue eyes and an allele for brown eyes. Now, just before eukaryotic cells divide, a number of characteristic thread-like structures become visible in the nucleus, and they stain quite easily with a particular stain. So chromosome, chromo means colored, and some means bodies. So this shows you a typical structure of a chromosome. Here it's made up of a pair of chromatids that are joined together by a centromere. This centromere can be found anywhere along the length of the chromosome. So after duplication, the chromosome comprises a pair of sister chromatids joined together by a centromere. The chromatids are identical to one another. Here you can see a simplified diagram of chromosomes that appear as threads under the electron microscope. If you unravel the DNA that makes up the chromosome, you'll see that it's wrapped around proteins, which we call histones. We call it a nucleosome when it wraps around 8 proteins. And then, of course, the DNA is a double helix. So this is looking at the chromosomes in more detail. Each chromosome contains one DNA molecule. This is highly coiled and held in position by proteins called histones, which you can see represented by those yellow circles. Together, the DNA and histones is called chromatin. The chromatin has a beaded appearance due to the presence of nucleosomes. And do you remember what I just said a nucleosome was? It's when you've got DNA wrapped around 8 histones, 8 proteins. So here you can see a typical structure of a nucleosome. DNA wrapped around 8 histones. Now, did you know that the total extended length of DNA is about 2 meters in human cells? But this has got to fit in a nucleus, which is very, very small, only 5 to 10 micrometers. So how does it do this? Well, the DNA is highly wound and tightly coiled. This is pretty cool. As you can see, the nucleosomes look like beads on a string. Okay, what's the difference between euchromatin and heterochromatin? And what is chromatin? Well, together, DNA and proteins, which we call histones, form chromatin. So as you can see here, the heterochromatin, which is that darkly stained chromatin, is tightly packed form of DNA, and it stains darkly. And where can you find it in the nucleus? Well, it's found on the periphery of the nucleus. Then euchromatin stains lightly, and the DNA histone complexes are loosely formed. So the DNA here is being used in protein synthesis. So heterochromatin is when you have DNA and histones that form a compact spiral, it stains darkly. Euchromatin is when the DNA and histones are loosely formed, and this DNA is being used in protein synthesis. Now in prokaryotes, they only have euchromatin, they don't have heterochromatin. Okay, so what is a gene? Well, we've all heard of the term gene. This is a section of DNA that codes for a particular protein. The position where it's found on the chromosome is called the locus. Here you can see the different coloured regions represent the genes. Now if you zoom in even further, you can see that genes have a specific sequence of base pairs of nucleotides in the DNA. Now you do get different forms of genes. For example, blue eyes and brown eyes. What do we call this? We call it the alleles. Okay, so what is a locus? The locus is the position on the chromosome where the gene is found. So here you can see a chromosome with different genes. And where the gene is found is called the locus. Notice how the first gene, which enables kidneys to retain phosphate, if there's a faulty expression of this gene, it can result in a disease, for example, a form of rickets. So you can have a look at the different genes and what happens if there's a faulty expression. And this chromosome is the X chromosome, so the female chromosome. Okay, what is a homologous pair of chromosomes? These are a pair of chromosomes that have the same structure as each other. One pair comes from the mother and the other comes from the father. If you look at this diagram, you can see homologous pairs of chromosomes have been lined up. The genes on each pair correspond to each other. So as mentioned earlier, genes have got different forms. For example, on the first pair of homologous chromosomes, you've got allele A and allele little a. So these will be the same gene, but different versions. For example, you could have allele capital A representing the allele for brown eyes, and allele small a representing the allele for blue eyes. So what is an allele? An allele is a different form of a gene. Now, if you put all the matching pairs of homologous chromosomes together, you get something like this. The pairing of chromosomes is called a karyotype. This is prepared by cutting out individual chromosomes from a picture and rearranging them. In this picture or diagram, here you can see matching pairs of chromosomes, which we call homologous pairs of chromosomes. Each pair is given a number. So in the original zygote, one of each pair comes from the mother and one comes from the father. There are 2 chromosomes that are the sex chromosomes. Can you guess which ones these are? Yes, the X and Y are the sex chromosomes, and all the other chromosomes are called the autosomes. So how do they prepare a karyotype? Well, what they do is they take pictures of the chromosomes and then they cut them out and they arrange them in pairs according to size and banding patterns. So karyotypes are used to determine if there's an abnormality in the chromosome number or structure. So let's have a look at the different errors that can occur in karyotypes. What can you see is happening in this karyotype? It's missing a chromosome. What about here? It's got an extra X chromosome. And here, chromosome 21 has got an extra chromosome. So we call this Down syndrome, or it results in Down syndrome. Okay, you've got an X and Y chromosome, which are your sex chromosomes. Which do you think is the X chromosome in this picture and which is the Y chromosome? The Y chromosome is smaller. So in females or humans, the females have got 2 X chromosomes and males are X and Y chromosome. Okay, let's have a look at the difference between haploid and diploid. Now, cells which have homologous pairs of chromosomes are called diploid cells. This is represented by 2n, where n is the number of chromosomes in one set. What about haploid cells? So not all cells are diploid. An example would be gametes. They've got only one set of chromosomes. So a cell which contains only one set of chromosomes is called haploid. This is represented by N. So how many chromosomes are there in a diploid cell in a human? There are 46 chromosomes. What about in a haploid cell? There are 23 chromosomes. So what happens if chromosomes are not halved at meiosis? So what happens if we don't get haploid cells? Well, you get all the genetic information coming from mum and all the genetic information coming from dad. So this would result in a child with too many chromosomes. So you need meiosis. And meiosis results in haploid cells coming together so that eventually you get the correct number of chromosomes in the zygote or the baby. So why can't a cell just keep increasing in size? Well, cells need to keep their surface area to volume ratio large. So what they need to do is they need to divide so that this surface area ratio remains large. So as soon as the surface area to volume ratio starts becoming small, cells need to divide. Here are a few questions which you can copy down and then answer in your own time. What does haploid mean? How many chromosomes are there in a human gamete? What does a karyotype show? And what's the difference between heterochromatin and euchromatin? Okay, let's move on to the cell cycle. Now, all cells are formed by division of existing cells. So eukaryotic cells always divide in one of two ways. You've got mitosis, which is the division into two daughter cells that are genetically identical to each other and to the parent. And then you have meiosis, which is the division into four unique daughter cells, which have only got half the chromosomes of the parent cell. So all cells follow the same sequence of events between mitotic divisions, and this is called the cell cycle. So there are three stages in the cell cycle. You've got nuclear division, cell division, and you've got interphase. Notice how long interphase is compared to the other two. So as mentioned earlier, cells don't divide continuously, but they do undergo a regular cycle of division separated by periods of cell growth. This is known as the cell cycle, and it's got three stages. You've got interphase, nuclear division, and cell division, which we also call cytokinesis. So interphase occupies most of the cell cycle, and it's sometimes known as the resting phase because no division takes place. This couldn't be any further from the truth because interphase is a period of intense chemical activity. And it's got three parts. You've got the first growth, the G1 phase, the synthesis, which is the S phase, and the second growth, the G2 phase. We're going to have a look at each phase separately. So the first phase is the G1 phase, which is the longest phase. The organelles are produced and protein synthesis takes place. So some cells will specialize and won't divide again. So in the embryo, this lasts about 6 to 10 hours. The second phase is the S phase, where DNA is replicated. So during interphase, the DNA in the nucleus replicates so that each chromosome consists of identical sister chromatids, each containing one copy of the chromosomes DNA. So here you can see this happens during S phase. Then you've got the G2 phase, also known as the second growth phase. So here the chromosomes start to condense and new organelles are synthesized. So which organelles replicate? You've got the mitochondria replicating, chloroplasts, and the centrioles. The next phase is the nuclear division, meiosis, so it's the division of genetic material, and then you've also got cytokinesis, which is the division of the cytoplasm and organelles. So there are checkpoints that check whether the processes at each phase have been accurately completed before the next phase. So here you can see the different checkpoints. You've got the G1 checkpoint that checks the length of the G1 growth stage and the size of the cell. You've got the G2 checkpoint, which checks the success of DNA replication, and it repairs and starts division. And then you've got the mitosis checkpoint, which checks mitosis accuracy. So if we have a look at what's happening during interphase, you can see that the DNA replicates, the centrioles, if present, replicate, and the cell prepares for division. So this graph illustrates how the quantity of DNA within a mammalian somatic cell varies during the different phases of the cell cycle. So notice how the quantity of DNA increases during the S phase and then it decreases during mitosis. So what's happening during the S phase? Well, during the S phase, the DNA is replicated. Each chromosome comprises sister chromatids, so that's why you're getting an increase in the DNA. Here are a few questions that you can copy down. So what are the different events in the cell cycle? What process occurs at G1 and which is the longest phase? Okay, in summary, let's go through what we've looked at. We looked at what a chromosome is. So it's a structure made of DNA and histones found in the nucleus of eukaryotic cells. Heterochromatin stains darkly and euchromatin stains lightly. So euchromatin is where you find the DNA used in protein synthesis. What is a gene and locus? A gene is a section of DNA that codes for a particular protein, and the locus is the position where it's located on the chromosome. What is a homologous pair of chromosomes? It's a pair of chromosomes in a diploid cell that have the same structure as each other and has the same genes. Not necessarily the same alleles though. What is a karyotype? It's an organized profile of chromosomes, and the chromosomes that have homologous pairs of chromosomes are called diploid. So they have two complete sets of chromosomes, whilst haploid have one complete set of chromosomes. And then we looked at the cell cycle. So the cell cycle is the complete set of sequence of events that takes place from one cell division to the next. And you've got 3 phases. Can you remember what those are? You've got interphase, nuclear division, and cell division, which is also called cytokinesis. And that concludes our lesson. The end.